a microsporal film. Microsporal film. Inside of the microsporal film is a microsporangium. Okay, and so the events that happen inside of one of those, and this is a close-up of the microsporal film. This is a longitudinal section through one of these cones. So this is not the cone. That's a cluster of cones. It's a bunch of them. This is one cone right here, okay? And so it has a central stalk, and swirled all around it are these modified leaves that are called microsporal phylls. Okay, when you see that the word P-H-Y-L-L, that suffix is always referring to leaf or leaf-like, all right? So modified leaf, the whole thing is a microsporal phyll. Inside of it is a microsporangium, okay? And so the events that happen there, first of all, in the microsporangium, is that you have a microsporal site. That is diploid, okay? Because this is a sporophyte structure. This is a sporophyte structure, okay? And it's there that this will undergo meiosis to produce 4 microspores, which are haploid, okay? And then they germinate. So when I say germinate, it means that they divide by mitosis. A couple of times to produce before they're released, Kerba can't draw it very well, is pollen. Usually it's about 4, 6 cells. So this is still haploid, but pollen is the male gametophyte. Right? Okay, now 2 of these cells, one of them, tube cell, is going to play an important role a little bit later on, and a generative cell. But this is all that's left of the male gametophyte. It is very much reduced, very much reduced. Okay? All right, so that's close up. You should have seen that yesterday, okay? By this time, what we have here is these are now pollen. And it's at this stage, that there will be a slit that will, as it dries out, there'll be a slit down at this end. In other words, it splits apart so that the pollen is released. So the pollen is released, okay, out of, and it's wind blown. Okay, and we're talking about millions, billions of pollen grains, and they're going everywhere randomly, wherever the wind happens to propel. So those sort of wing-like things on the side, I already said that. Okay, maybe gametophyte. It's also male gametophyte. I'm going to stay with that microgametophyte. Okay, so this is pollen grain that you saw. These sort of wing-like cells, they help it, the pollen be wind blown, you know, like an airplane, like the glider. However, this pollen does not go very far. Okay, it's if you go out to any of these conifers around here, you mostly find that yellow pollen grains very close to the tree. Okay, so if it happens to land on a, okay, I've already said that, already said that, already said that. If it happens to land on a female cone, ovulate cone, okay, that is called pollination. So if we have here ovulate cone, the ovulate cones are, of course, much larger, all right? And I want to really emphasize that this here is producing pollen is a big expenditure, energy expenditure on the part of the plant, okay? So this is now the ovulate cone. These are the big cones that you normally see. Those are the things that you normally see on the tree and call them pine cones. Even if it happens to be a fir tree, you still call them pine cones, but no, it should be just called fir cones, all right? All right, so a longitudinal section down here. what it has here is a megasporophyll, okay? So one of these is a megasporophyll, and I'm going to zoom in on it, I think. Well, let's see here. Okay, let's back up. Actually, let me go to I'll come back to this in a moment. Okay, so this is zeroing in on one megasporophyll, which is, of course, one scale. Okay, if we back up here to this picture, one of these scales happens to be the megasporophyll. Now, when it's early spring, these scales are real loose, and there's a lot of sap in between here. You pick these up, they're very, very sticky. So if this pollen happens to fall, if it happens to fall right on here, then it gets into that sap, and it is drawn in between the scales. It is drawn in between the scales toward the center of the cone, okay? It's gone there into the center of the cone, so it would land out here somewhere. Actually brought something that I can point with, because it's too far. Don't have a laser pointer, but it lands out here, and eventually, because of that sticky sap that's in here, as it gradually dries out, the pollen will be moved all the way in to right in this area here, okay? And that process is called pollination. Pollination is simply the capture of a pollen grain onto the female ovulate cone. That's pollination, all right? Now what happens here, this part here, is this here is the ovule. Okay, so part of this ovulate cone, part of the megasporophyll, happens to be something here that is called the ovule. Okay, that's the ovule. In fact, let's see if we can get a drawing on this. So this whole part there is the ovule, and some at the very outside of it, I can't write on the wall way up there, it's too bad, but this part right in here is going around the outside all the way around. It's called the integument. Okay, it's hard covering. Okay, it's rather woody. And then in here, there is material that's called nucellus. Okay, nucellus. That will be eventually part of the food supply for the seed. Now, what happens here, so this is the megasporangium here. And so what will produce, this is not shown here, and I don't have any slides of it, but it will produce one cell in here that is the megasporocyte. Of course, it's diploid, okay? And it parallels that same thing up there, okay? This will undergo meiosis, produce 4 megaspores, which are haploid. And of those 4 megaspores, one of them is larger than the others. And these other 3 will atrophy. They just disappear. They die.
What produces the megasporocyte?
What's that?
Like
This- the original big cell, megasporal site, and then after it undergoes meiosis, they become megaspores.
Okay.
Yeah.
So the pollen's not involved there, right?
What's that?
The pollen's not involved there?
The pollen at this time has already been deposited over here. Yeah, so this is happening during, fertilization hasn't taken place yet, okay? Because it takes usually a year or longer, okay? All right, so you have there this one surviving megaspore, then germinates. To become female female gametophyte. And she will produce she will produce 2 to 3 archegonia. Okay, now this female gametophyte is relatively big, you know, but it's still setting in this ovule. Okay, it's located inside of the ovule. Yes.
Is the ovule where the pollen lands?
The pollen lands in between the scales, and it will come down here. This is called a pollen chamber. Pollen chamber, but it's still outside of the ovule. Now there will be an area here of weakness that's called the micropyle. It's not shown there. Through which the pollen will eventually grow. Okay, but we'll get to that here in a moment. All right? So I've already gone through this. But let me pull this down. Don't need that anymore. All right, yeah, so here, I've already drawn that up on the board. We have the megasporal site meiosis, only one of those spores survives, mitosis, about 2,000 cells. Now, so the female gametophyte is much larger than the male gametophyte. The male gametophyte, only about 4 to 6 cells, that's all. And it is still in the ovule, and so what we have here, this is the female gametophyte, right there. And the micropyle down here. Okay, so this is the center of the cone. Right here is the center of the cone. This is the outside of the cone, outside of the scale. And so the female gametophyte is lying right here. The pollen grain is right over there. And here we have 3 archegonia. And so this archegonia, the cells around it that form it are very small, but it produces one real big egg inside. Just one egg in the archegonia. Okay? All right. So this is the female gametophyte. And then what happens here, we'll skip this for a moment. Okay, I already said pollen I already said all this. So that pollen grain happened to be now in the pollen chamber. What will happen, we had 2 cells, I said, that were important there, the tube cell, generative cell. The tube cell starts growing a tube called a pollen tube that will digest its way through the micropyle. It's being guided by chemicals that are released by the archegonia. And so it's being directed toward the archegonia. So it will grow in that direction. And that's the micropyle. At the same time, this generative cell, it divides. And one of those, and this is not a cell now, well, it is a cell, but one of the nuclei, one of the resulting 2 nuclei that comes from that division, what type of division is that? Mitosis or meiosis?
Meiosis?
Try it again.
Mitosis.
Oh, what did you say?
I said meiosis.
You said meiosis?
Mitosis.
What do you think?
Can you
This is
repeat
the gametophyte.
the
What is the point of the gametophyte?
Oh, mitosis is acting.
She's in. Okay, so if she went through meiosis, we'd have half an in. That's not possible. Okay, it's mitosis. This is divided by mitosis. The sperm and eggs are produced by mitosis. Okay, contrary to what we have in animals. Okay, so one of those sperm nuclei is there, and it moves down the tube nucleus. So the tube nucleus has cytoplasm that it can move through, and it will move down, penetrate into the female gametophyte, where the egg is, and the sperm nucleus will be released into the archegonia. And then we get fertilization. Fertilization, and that produces the zygote. Okay, so just to go back here, this is showing the life cycle. So we're at this point. Here's our pollen. Here is the megaspore, but it becomes a female gametophyte. Down here is where the pollen grain is. In that pollen chamber, the pollen tube is growing and digesting its way through the integument and through the nucellus to one of these eggs. Even though there might be 2 or 3 eggs there, only one of them gets fertilized, and then the others will atrophy. Okay? And furthermore, the female gametophyte, now what will happen here is we have a zygote. It starts dividing by mitosis. This is now diploid, right? It's a fertilization event. Set of chromosomes from the sperm, set of chromosomes from the egg. Therefore, it's diploid, 2 sets of chromosomes. And it will become a baby sporophyte. Baby sporophyte, okay? The embryo. And it will form a seed. The ovule becomes a seed. The ovule will become the seed, where the integument is the covering of the seed. And what the seed consists of is the embryo, diploid, and food reserves. Now, the food reserves are in part, the gametophyte. The remnants of the gametophyte serve as food. Furthermore, I mentioned that there was another material in there called nucellus. That is also serves as food reserves. Okay? And then eventually this will be released. When the seed falls on a suitable place, of course, it cannot become photosynthetic until it sends up leaves, and it takes some time. And this is the reason for the food reserves. That provides the carbohydrates, the amino acids, for cellular respiration and so forth, and also for building the materials of the plant until it gets big enough to produce its own photosynthesis. Now, in the non-seed plants, how this was accomplished was by having the sporophyte initially attached to the gametophyte. The gametophyte is photosynthetic. Okay, that's how it gets there. All right. So in this seed, there are 3 different generations of tissues. The seed coat and the nucellus came from the original sporophyte, that first generation. Some of the food reserves is the remnants of the female gametophyte, second generation. And then we have the 3rd generation, namely the baby sporophyte. So so what would be the ploidy of the cells on the seed covering? Haploid or diploid?
Diploid?
Diploid, because that's part of the original sporophyte. That was part of the original ovule. And then some of the food reserve is haploid, some of it is diploid. The nucellus part is diploid, the female gametophyte part of it is haploid, and so forth. And of course, the baby sporophyte is haploid. And that completes that life cycle. All right, so keep it straight that pollination, fertilization are not the same thing. Pollination and fertilization are 2 different events. All right, and I think, so the seed, already said that. Now, the embryo itself consists of a tip that's called the radicle. The radicle will become the root. And then it will have anywhere from what are called 3 to 12, depending on the species of the plant, cotyledons. Now these cotyledons, what is the function of the cotyledons? They are not leaves. They are not future leaves, although that part of the embryo will give rise to the shoot part that has leaves. The function of the cotyledons is to mobilize, they have enzymes that mobilize the material in the food reserves, okay? Furthermore, these things produce hormones, growth hormones that stimulate the proper cells within the embryo to grow, okay, and grow in the right direction. So if I go back here to that picture, actually I guess it's, where was it? Right here. These up here, those are the cotyledons. And this down here is the radicle. This will become the root. And these, of course, from this part up here, they'll eventually produce the photosynthetic leaves. Okay, that's the life cycle of conifers. So that's what the seed is, the embryo plus food plus a seed coat. Now for pines, this life cycle will take, depending on the species of pine, 2 or sometimes even 3 years. So it's a long life cycle. But these trees are also usually very long-lived as well. Okay, so here's a pine seed. If you cut it open, it's the radical right here. That's the embryo. Those are the cotyledons. And this is the food reserves. It's necessary for the plant to get its start in life. Okay, and I already showed that. All right. Any questions about the life cycle conifers? They're similar. It's similar in the ginkgos. It's also similar in the cycads. Okay, but we're concentrating on the conifers.
Can you go back to the seed slide?
Which one?
The seed slide, the 56,
This one?
the one before it.
This one? You want me in the picture too? I'll smile.
Can't see.
Okay. All right, any questions on that? So you see, this is still alternation of generations. Now, for the next spermophyta is angiospermum, okay? And I'm not gonna draw the phylogeny, but you guys should be able to do this. Incidentally, for exams on Monday, the exam will be on Monday. You'll have questions similar to what I had on this last quiz. In fact, a lot of the quizzes, all the quizzes, you'll have questions like that. You will have a data set for building a phylogeny. And that phylogeny may have, or the data set may have character conflict. Okay, it's not gonna have massive amounts of character conflict, but there may be 1 or 2 characters that are in conflict with the general trend of the data, all right? So beware of that. You will have questions. You really need to know all the phylogeny, the parts that I have presented here, and at least 2 synapomorphies for each clade if 2 were given. Sometimes only 1 was given, and in some cases none at all, okay, for example. And you may have to draw a phylogeny. I might say, draw the phylogeny of all life up to the level of the supergroups. Okay, or that's going to be hard to tell you where to quit on that. So what I might do is I might draw a phylogeny and leave all the names and apomorphies off. And you have to put in the names. Okay, now on some of these groups I've given, sometimes up to 5 or 4 synapomorphies, but usually on an exam, I only ask you to give me 2 at most, to keep it simple. So, give you some idea there. So, I'm not going to draw the phylogeny here, but yeah, let's go ahead. Just to remind you of plants. In fact, let's just put it back here. I don't want to eat up too much time because I'm already far behind as it is. But for the RT class, we go. What branches are first? Vote off of it. And then what's the name of this place? And then, what would I have right there? We'll ask for any synapomorphy for simple reasons, paraphyletic, right? Okay. You can only have synapomorphies for things that are monophyletic, right? And then what I have up here. Okay. If we put something like I did at the beginning of this lecture, and then plant. Then what do we have? Bryophytes. And then what's this called? Okay, Tracheophyta. Okay, that's one I don't know how to spell it. I think we put it there, so we can tell. And then, in this, we have GKO, I think it is. Then this one here. And then the last one we're coming to down here. And yes, sir. Good. Okay, yes.
What's the additional blade for carotid?
For,
Carotid. Oh wait, how many moves do we have? Sorry.
is here?
No, the one next to it. I thought that was carotid.
Well, since it's paraphyletic, I'd like to do two lines or something like that, or even like that, you know, the thing is, it's not monophyletic. But I would probably on it, if I gave you the phylogeny and you were filling in the names, I probably wouldn't do this. I'd probably just have one line up like that, but it's not monophyletic.
What about Bradford?
What's what?
Bradford.
Bryophytes, moss, liverworts,
Oh, yeah.
okay, the non-vascular plants, okay. All right, okay, so, yeah, you got that down, just wait till we get to animals. Okay. All right. Maybe I'll just leave that on the board for the next class that comes in here, or really freak them out.
Is Rhodophyta its own clade under Archaeoplastica?
I'll say what?
Is Rhodophyta its own clade under Archaeoplastica?
Oh, yeah, it's an archaeoplast.
And then it's separate from Viridiplanti,
For the,
right?
yes, that's right.
Okay.
Because the one from Viridiplanti, those are the green branch, and Rhodophyta is the red branch.
Oh, okay.
Okay, all right? Okay, so going on with this. Wake up. Okay. Come back. Where'd my display go? Got you, walk away from it, and it's fickle. Make me do this again. Okay, so for the angiosperms, these are the flowering plants. And although the names of some of the features and life cycle are going to be a little bit different, they're going to be very, very familiar story as to what you had with conifers. Very, very similar to that. They also go with pollination, and pollination is the same thing. The movement of the male gametophyte pollen from the male sporangium, called anthers, to the female part of the sporophyte where the egg is going to be produced. And so that's what, now here pollination, one of the advances of the flowering plants is that they have evolved a relationship with various other types of organisms, mostly insects, bees, ants sometimes beetles, but also hummingbirds and sometimes even bats as pollinators. And therefore, their relationship with these pollinators means that it's economical for them, because now they don't have to produce tons and tons of pollen, okay? Because now it's more directed, because a bee will come to a flower, and as it's trying to get to the nectar at the base of the flower, it'll rub against the anthers, pick up pollen, and usually they go to another flower of the same species. And therefore, it's more direct delivery of the pollen. Therefore, this pollen tends to be sticky, okay, and it's not real small sometimes, and it doesn't get in the wind. Now, there are some flowering plants that do have wind-blown pollen. And those that have wind-blown pollen, they have to produce a lot of it, okay? They have to produce a lot of it, and usually these have very small, green, inconspicuous flowers, okay? Because you normally don't see flowers on grass very much, do you? But they do produce flowers. They're part of this system of angiosperms. All right, now here, the gametophytes are even more reduced in size, both the male and the female. Furthermore, their vascular tissue is more refined. What we mean by this, they have tracheids. That's true of ferns, sporophytes. That is true of conifers and gymnosperms. But angiosperms have tracheids also, but from tracheids that has evolved something else called vessel elements. And we're certain that these have evolved from vessel elements, or vessel elements have evolved from tracheids. And these are more efficient at conducting water, okay? Because these two types of cells, tracheids, vessel elements, they're found in the xylem. The xylem, part of the vascular tissue. That's wood, okay? All right, and that's an apomorphy. That's a synapomorphy. I have those double stars there. Furthermore, they have reproductive structures on the sporophytes called flowers. That's also an apomorphy. Just like the cones are a collection of modified leaves, that's also the evolutionary origin of flowers as well. They're highly modified clusters of leaves. Furthermore, there's another innovation that the angiosperms have, fruit, okay? Now the fruit is the ovary of the flower will become a fruit. The ovary of the flower will become a fruit. It contains the seeds, and it's a dispersal mechanism. With the conifers, when they produce their seeds, on the cone, the ovulate cone, the scales will open up and the seeds fall out. They can be windblown a little ways, but they don't go very far. But here with the fruit, it's a way to really spread seeds, disperse them way, way away from the parent plant. And we'll look at those as we go through here. All right, so this is just showing cross-section of the xylem with tracheids, okay? These small cells are tracheids, the large ones, diameter, these are vessel elements. So this is what the tracheids look like. They're usually long, tapering types of cells, perforated with little pores so that water, and they're hollow, they're dead at maturity. All that's there is just the cell walls, okay? And so it's like a tube, and water can move up, and where the two different tracheids overlap, there is a lot of pores in the cell walls that allow water to go from one cell to the next. These vessels are larger, and they're not as tapered. They are tapered, but they stack one on the other, and they're more efficient at carrying the water. Furthermore, they have a lot of strength because lignin in the cell walls. All right, so life cycle, we'll go over it. Heterosporous, just like in the conifers, meaning that we're looking at two different types of spores that get produced. Flowers are structures with sporophyte and consist of, actually the flowers consist of a whorl of modified flowers or modified leaves, and there are 3 different whorls. In fact, I'll show them in a moment because it's on the slide. Part of them, flowers, they have stamens. Stamens contain, so this is a highly modified leaf. At one point in the stamen, it's called the anther. And I'll show a picture of the flower here in a moment. That is where the microsporangia happen to be. That corresponds to staminate cone sporangia, okay, in the microsporophylls. The female part of the flower is called a carpel. It's also special modified leaves. At the base of the carpel, there is an area, it's called the ovary. And inside the ovary will be multiple ovules. And I'll show this here in a moment. All right, yeah, here's the flower. Okay, so I said that we have a stem here. We have one whorl of leaves. Then further down the stem, and I'm making this real primitive, showing evolutionary origin. And then one up here. This first whorl of leaves has been modified to become sepals. Sepals. Okay, now the collection of all sepals, now the sepals are the most leaf-like, and they are photosynthetic, okay, but they're at the base of the flower. Sepals, the whorl of all those sepals, they're special names, we don't need to worry about them, okay? I don't want you to worry about them. Just only the sepal, that's this first layer. And then the next are the petals. Usually the petals are colored, okay, they're very colorful. And not the sepals and the petals have nothing to do. They're not fertile material. Okay, it's not fertile material. It's sterile. The petals are very important in attracting pollinators. Okay, in addition to the smell, that also attracts pollinators. The next whorl, the third one, these are really modified leaves. They're stamens. Stamen consists of the anther at the end. The anther usually has 4 chambers. 4 chambers, 4 sporangia. So inside of the anther are the sporangia. Okay? And then lastly, right in the middle is the carpel, which consists of the last whorl. Now, primitively, there were separate, okay, the carpel is a fused structure of things that are called pistil, P-I-S-T-I-L. We're not going to use that word, but in some magnolia type of trees, flowers, they have separate pistils and they're not fused together. But in most of the angiosperms, they have fused together into a single structure called the carpel. Okay, so here in the carpel has place where the pollen has to be deposited, called the stigma. Then there's a part called the style, down at the base of the flower. So if we took cross sections here, these sepals would on outside, the petals intermediate, stamens connect next, and then we would hit the ovary. Inside of the ovary are areas called ovules. Okay, all right. So this here, the ovule, is pretty much analogous to the ovule in a ovulate cone. Okay, has the same correspondence function. Okay, all right. Oh, something more complicated here. Yeah, there's in the literature you'll come across these words quite a bit. Perfect versus imperfect flowers, and another terms here, perfect versus imperfect, complete versus incomplete. I don't have that up there. What this perfect and imperfect is referring to is whether or not the flower has the male or female parts. It's perfect if it has both stamens and carpels, both the female part and the male part. Okay? That's a perfect flower. If it's missing one or the other, that's an imperfect flower. And it can either have staminate flowers, where it just has the stamens, but not the carpel, or it can be a carpellate flower, where it has the carpel, but not the stamens. Now, one interesting little thing of complexity in flower structure is that one particular plant, one individual, may produce both staminate and carpellate flowers. Okay? The same individual, different locations, have staminate flowers, carpellate flowers. Other species may have individual plants that have staminate flowers only, and other individuals of the same species just carpellate flowers only, okay? But usually what you have is perfect flowers. You go out here, pick a lot of the wildflowers growing around here, most of them have both carpels and stamens. Another two words, which you'll get in lab, so I'm not going to really talk about it too much here, would be other than perfect, but complete and incomplete. Complete is if it has all four flower parts. It's incomplete if one of them are missing. Okay? So a staminate flower, by definition, is incomplete because it's missing the carpel. A carpellate flower is incomplete because it's missing the stamens. We may have a flower that is perfect but incomplete because it might be missing the sepals. Okay? And so flower structure, flower terminology sometimes can get very, very complicated. All right? Two other words. This we'll be using a lot in animals. Okay, so I'm going to introduce it here. In animals, okay, their reproductive structure, they're monoecious if we have individuals that have both ovaries and testes. In other words, they have both the male component and female component. They produce both sperm and eggs. That's monoecious. And we're going to see some animals like that, like earthworms, for example. All right? Dioecious means that you have separate sexes. Okay? Males that have testes, females that have ovaries and produce. But these terms can be applied to plants as well, and it gets very, very complicated. Because if we have an individual plant, because it once again depends on the species, that the individual only has staminate flowers, then it is dioecious. But if we have a plant that has both staminate and carpellate, then it is monoecious. Okay? Gets complicated. Think about that. You have a whole weekend to think about that. All right, so going on here. Microspore production in the anthers. And it's a similar story, very similar. Up here, that's this anther, that's where the sporangium happens to be. And it produces, from the sporophyte, microsporocytes, which undergo meiosis to produce microspores. The microspores germinate by dividing once. And they do, instead of having 4 cells, they have 6, which is 2. A tube cell and a generative cell. That's all it is. And that's the male gametophyte. And all that story happens right up here in the anther. And when the pollen is mature, the walls of the anther split open so that the pollen can be released. Very easy. Okay, so that's what's happening here. Meiosis, microspores, they're usually in a very thick wall. Mitosis, just one division. And they consist just of a tube cell and a generative cell. That is an adult plant, the gametophyte generation. The whole plant is just 2 cells. So the story's just like in staminate cones, just in a different location, that's all. All right, everyone have this? Going on to the next. Pollination, okay? So with pollination, simply removal, the pollen is released, and it has to find its way to the stigma of the flower. Best of all, if it's on a different plant, different individual. Sometimes that can be on the same flower. However, now, those that use pollinators to move their pollen from the anther to the carpel of another flower usually are bright colors. Okay, bright color, because it attracts the pollinator. So birds, insects, because when the birds and insects are coming, they're not coming to eat the pollen. At the base of the flower, down near the ovule, there are glands that are called nectaries. And they produce a sugar substance, and that's what the insects are coming from. I'm not sure if that's you, but that is what they're coming from, nectar. And, but this is what, they'll pick up the pollen. By way of getting to the nectar, they rub against the anthers and the pollen gets on them. And then when they go to another flower, try to get the nectar. Now, these flowers never produce enough nectar to satisfy the pollinator. Okay, they just get a little bit. Because if it's completely satisfied the pollinator would go away and take a nap. And therefore, the pollen wouldn't get moved from one flower to the next. And so there's been a trade-off here. Now, those that have wind-blown pollen, they don't have colorful flowers, right? And they're inconspicuous flowers, small, green, usually produce lots of pollen. And this is what gives you hay fever, okay? Along with fungal spores, they can cause hay fever as well. But this is where you get an allergic reaction to this type of pollen, not this. Which irritates me a little bit, because you know, I remember back in the days when I watched TV, I usually don't watch TV, but they'd have advertisements for certain types of allergy medicines. They would show this young lady frolicking through this colorful field of flowers and sneezing her head off. Now, to me, that was ridiculous, because you're not getting allergies from that type of pollen, not from bright colored flowers. She should have been on that advertisement running through a bunch of ragweed, okay? That would make sense. Anyway, my wife doesn't let me watch TV. Okay. All right, so large quantities are released and so forth, and this is what contributes to hay fever, which is an allergic reaction. Okay, now, so we've seen what happened here with the male forming the pollen, but in the ovaries of the carpel, ovules, okay, first of all, pollination, this is what happens, is that pollen will arrive on the stigma, and the tube nucleus will start growing. And it's digesting away down through the style of the stigma. And it's guided by, because in the ovary, in the ovule, there is the female communophyte, and we'll find out where she came from here in a moment, that is producing chemicals that are attracting the growth of the pollen tube. So the pollen tube is breaking between cells, disrupting, and moving down. In fact, it looks like this tube over here. Okay, so pollination takes place when pollen grain gets there. Tube cell starts growing, and it's tracted down toward the ovule, which has the female gametophyte. Now, let's look at, first of all, where this female gametophyte comes from. And now this is only showing this whole area here is one ovary, but for simplicity, they're showing just one ovule. When you looked at the lilies yesterday, cross-section, there was 2 ovules here, 2 ovules here, 2 ovules there, 2 ovules there. And if you look at something like a flower for tomato, there would be hundreds of ovules. Okay, each ovule. Let's go back here and see if I'm missing. This doesn't show it either. Okay, I'll just draw it on the board. So in that ovule, first of all, micropyle, okay, that's the micropyle. It would have, first of all, mega sporal site. 2 ends. Similar story to what happens in the ovule of a ovulate cone. This will divide by meiosis, so that in that ovule, 4 nuclei, one larger than the other. So these are the megaspore. And 3 of them die. Only one survives. This will then divide. It'll divide 4 times, okay, and actually 3 times. 3 mitotic cell divisions that will result in the mature female gametophyte. 8 nuclei, 7 cells. 8 nuclei, 7 cells. Okay, so when the megaspore divides first, we have a female gametophyte consisting of 2 cells. They divide again, 4 cells. They divide again, 7 cells, 8 nuclei. 8 nuclei. Okay, so they will have, down where the micropyle happens to be, 3 cells, right there. One of them is an egg. The other 2 cells are called synergids. The synergids represent the remnants of the archegonium. Remember, archegonium, that's where you produce the egg, right? Okay. In the middle, there is one big cell called a polar cell that has 2 nuclei. Okay, we don't call that diploid. Okay, that's not diploid because each cell or each nucleus is haploid. Okay, just binucleated. That's all. And then at the other end, there's 3 cells that are called antipodals. And they represent the remnants of the non-reproductive parts of the female gametophyte. So the female gametophyte has been reduced from 2,000 cells in the conifers down to just 7. Okay, that's what's happening here. So if we go back, follow our story. As the pollen grain is growing, tube cell, the generative cell divides by mitosis to become 2 sperm nuclei. Okay, so our pollen had 2 cells. Tube cell, it starts growing the tube, pollen tube. The generative cell divides, and we have 2 sperm nuclei. 2 sperm nuclei. Now, obviously, this tube, pollen tube, has to be built from carbohydrates, proteins, etc. Where's the material coming from to build the carbohydrates and the material of the tube? Pollen grain doesn't have, it doesn't eat. So where is that sugars and proteins coming from to build the pollen tube?
From the remnants,
From the what?
from the synergies.
Well, it's coming from the carbo itself. I mean, this in here is producing that material that can be taken up by the tube cell. Okay, so it is that carbo that's providing the material. Anyhow, what happens is now the tube cell, here's the micropyle. The tube will penetrate the micropyle, and both sperm nuclei will fertilize. One sperm nucleus fertilizes the egg. That's one fertilization event. The other sperm nuclei will fuse with the polar cells. All 3 of those cells will go together, and we have a triploid entity. 2 fertilization events. Okay? One produces the zygote. The other one produces something that's going to be called endosperm. Okay? So, double fertilization. This is a synapomorphy of the angiosperms too. So we have a lot of apomorphies for this particular group. So, as this develops further, we have this double fertilization event, and as we develop further, where am I? Over here. Okay, so this is a good picture of this is an ovary, and you see it has 1, 2, 3, 4, 5, 6 ovules. Okay, so don't interchange ovary and ovules. They're separate things. Okay, but once we get the fertilization, double fertilization, then what will happen is that the egg, the zygote, will develop into the embryo sporophyte. It's diploid. The endosperm divides, and becomes what is called endosperm. And the endosperm is the food reserve, and it's triploid. Okay, 3 sets of chromosomes. And it is triploid. Now, why this evolutionary step to provide endosperm? If you looked at the conifers, there was a lot of food reserve there from the nucellus and also from the female gametophyte. But how big is the female gametophyte here? That's not much to eat. Female gametophyte's pretty small, just only now left over, 5 cells. That's not enough nutrition to grow the plant. So this is a way that angiosperms have come about to form the food reserve, and then we have a seed coat. Okay? So what we have here, the ovule becomes the seed. The ovule becomes the seed. Okay, so this is showing just the zygote. We're not going to worry about this, but by the time we get to the end, we have the ovule has a seed coat, and there's a radicle. Okay, that's the root I picked, calls it same word as I used before, radical, and it's embedded in the endosperm. The endosperm is the food reserves, and the embryo here in angiosperms will have either 1 or 2 cotyledons. 1 or 2 cotyledons, that's all. And the cotyledons have the same function as what I mentioned for the conifers, is that they produce hormones, growth factors. They also control the breakdown of the proteins that compose the endosperm, because they're deposited there as full proteins, storage proteins, and they're broken down to amino acids so that the embryo can use them and so forth. And that's the food supply. All right? So, you like to eat peanuts? I love peanuts, okay? So, you break them apart, and you know that little knobby thing at one end. How many of you have experienced that? That's the embryo. The rest of it, the 2 sides, are the cotyledons. Okay? The embryo is real small. Okay, so we're not going to worry about these words, hypocotyl. I just want you to know radical and cotyledons. So this is bean. So our baby sporophyte is just there. These are cotyledons. They provide food. This plumule will eventually become a photosynthetic leaf when it grows up. But in lab, we'll look at this in more detail. Okay? Now, that's where the seeds come from. So you see the story here between conifers and angiosperms is very, very similar. Just only different structures.
So the spermatocytes growing out of the radicles?
No, the radical will become the root. It's growing, what will happen is that it will start growing, and the plumule will come out of the soil, and the radical will bury it.
And the cotyledons on the other side?
It depends. The cotyledons will be on some, 2 different forms here. Sometimes the cotyledons are down in the ground in some of these plants, and the plumule will become the shoot. In other cases, the cotyledons are above ground, but eventually they disappear because they're just food reserve. They don't become leaves. Cotyledons do not become leaves. Yes?
So the nutritive tissue and endosperm, just like.
That's in the sperm, yeah.
Oh, it's the same.
Yeah. That's where the plant is getting its nutrition. Okay, the ovule develops into seeds. The ovary becomes the fruit. So, 1 flower, 1 fruit. However, it might be 1 flower and many seeds. It depends on how many ovules happen to be in the ovary. okay? So this develops into the fruit, and this, this is sort of where that's the ovule, that will become a seed. This whole thing here is the ovary, and that will become the fruit. Okay? For example, tomato. Is that a vegetable or is that a fruit?
Fruit.
It's a fruit, okay? Is corn, is that a vegetable or is that a fruit?
Fruit.
It's a fruit. Now let me see here. Is celery, is that a
Vegetable?
Yeah, because that's a stem, all right? So this idea of fruit, what is fruit and what is vegetable is really a dietary concept. We like things that are sweet, call them fruits. And some are fruits, some are really not fruits, okay? This is a fruit. And all those seeds, each one of those seeds, tomato seeds, come from one ovule. But this here is the fruit. This aids in dispersion, okay? So with conifers, their seeds don't blow too far away from the mother plant. But here we can have some animal come along, eat this up, feel real good, run over this hill and over the next hill, and then all of a sudden stop and take a poop, and the seeds are there, fertilized, okay? Way, way long away from the mother plant, which means that the baby plant doesn't compete with the parent plant. Okay, so it's an advantage. Now, so, of course, the fruit is derived, so the ploidy of those cells, haploid or diploid? The diploid, because that is tissue that's produced by the original sporophyte. Okay, that's tissue from the original sporophyte. So in this fruit, we would have diploid tissue from the original sporophyte. Inside of the seed, we'd have diploid tissue that is the baby sporophyte. And we can forget about the gametophyte, it's just not there, it's not there at all. And even there in the seed, we also have the endosperm, which is the triploid. Okay? All right, various types of fruits. I'm not going to have you learn all these different types of fruits, but just pointing out here, in some cases, there are some flowers, for example, cherry flowers, peach, that'd be another one, that their ovary contains one ovule, because in the fruit you have just one seed. Okay? In other cases, we have, in fact, pineapple, that's from a cluster of flowers. And so it's not just one individual fruit, it's a multiple fruits that are put there together, and so forth. But just getting an idea here as to what is a fruit. And that's a fruit. That dandelion part, yeah, the seed is right there, but this here is the fruit. So not all fruits are sweet, and do you want to eat them, okay? But that still aid in dispersal. It still has that function, dispersal. And somewhere where I grew up, they have these things called devil's claw. Yes?
So the fruit doesn't have to enclose the seed?
It's connected to the seed, it doesn't have to enclose it, yes, that's right. Okay, the thing here is called devil's claw. Flowers are pretty nice, and it's not native to the United States. I think it originated in Central Asia, but it grows on the high plains. And when it produces a fruit from that flower, they look like this, and this is hard and woody, has these two long claws, that's why it's called devil's claw. Inside here are the seeds, and you see cattle walking around, and even dogs a lot of time, and even some people, that these things will grab onto you, and they're hard to get off, and that carry the seeds far, far away before they can ever fall. So it's still dispersal, still seed. Okay, all right, so that doesn't complete the angiosperms. The last thing here I'm going to add to the phylogeny is we're going down to what is angiospermy in the Linnaean system is the phylum. But there's two different, I'm going to put down here on the phylogeny. only two that's real important here, okay? There's some basal groups of angiosperms that really don't fit into real good clays, and so we're not going to worry too much about it. But they're at the base of the angiosperm groups. These are called magnoliids, and they're not. But we know that they're sort of plesiomorphic. But exactly the relationships to each other and to the two major clades of angiosperms, which are monophyletic. And one of these is the monocotyledon. Okay, this is monophyletic, and these are called colloquially monocots, but the scientific name for the clade is monocotyledone. And these are some synapomorphies that's unique to them. Their leaves, their vascular tissue that happens to be in the leaf, they're parallel. So the veins are running parallel. This beast, like palm, lilies, and all grass, happen to be monocots. Okay? Furthermore, in the seed, it only has one cotyledon. One cotyledon. An embryo with one cotyledon. That's synapomorphy. This is not an apomorphy at all. It's just something that is usually the case, is that their roots happen to be fibrous. So at the bottom, the roots don't have any main axes. So this is just only a major characteristic that frequently find with them. So palms, grass. Grass is a huge group. Okay, it shows up in the fossil record probably about 40 million years ago in the Cenozoic. And they have really, it's a huge family. Okay, it has many, many species. The other clade that is monophyletic is called eudicotyledone. Okay, now obviously monocot means one cotyledon. This means two cotyledons, but we have this prefix EU meaning true dicots, okay? Because they do have two cotyledons instead of one, but that's not an apomorphy. The reason it's not an apomorphy is because many of those magnoliids, they have two cotyledons. So that makes plesiomorphy, all right? So the only real good apomorphy that I could come up with, this group is well supported by molecular data, okay? Well supported as monophyletic, but they have what is called a tricoplate pollen. It means that it has three ridges to it, whereas the monocots don't have anything like that, but all eudicots have that. There are other differences between monocots and dicots that we'll go into a little bit later on, okay? Now, as far as generally the vascular tissue in the leaves is usually dichotomous branching. Yes?
Are the magnoliids, the monocotyledone and eudicotyledone, all individual clades? Like
No,
separate?
these are not clades. They're just sort of groups that uncertain relationships down the base of the angiosperm tree.
Okay.
So they might be paraphyletic. They are very unlikely to be monophyletic.
But the other two are unrelated?
These two are monophyletic.
Okay,
Yes.
but they're not under mono?
No. You would have, so these are three different groups, one of which is probably paraphyletic.
Mhm.
So I'm not, and it's a small group, we're not going to pay too much. But the monocots and the dicots, real huge groups, and they are monophyletic. So in these seeds, two cotyledons, but that's not an apomorphy, okay? Because the other group, the magnoliids, they have two cotyledons as well. Usually the roots are taproots, okay? So that there's a central axis to the root that goes down real deep, and then you have other subsidiary branches coming off of it. And later in the class, when we get into plant physiology, anatomy, we'll go into more of the details on that, okay? All right.